All right, the last unit that I want to cover in this class has to do with antimicrobial drugs, which I will start talking about today. And then after our exam on Thursday, we have one lecture day on the Tuesday after Thanksgiving, where I will finish up this topic and also talk about drug resistance. Because attitudes toward antimicrobial drugs are changing a lot, and resistance to those drugs is a huge societal problem right now. And I'm going to be relating these things back to other things we've talked about all semester. Okay, does anybody know who this is, or who this might be? I hope that didn't make it onto the audio. This is the person that discovered penicillin, who is Dr. Alexander Fleming. Dr. Alexander Fleming. And he was the person that discovered penicillin. And I don't remember if we talked about this in the beginning of the semester, but his discovery of penicillin was actually an accident. He was not trying to find it. He was not looking for any type of drug. What he was actually doing was studying bacteria and growing bacterial colonies on plates and then using them for other purposes. And he sort of had something frustrating happen. He kept getting fungal contaminants on his plates. And so instead of having a plate full of bacteria, he might have fungus growing all over the plate, and that was frustrating and irritating, and if it was me, I would just throw those away. But what he actually noticed was, if fungus was growing on a plate, there would be a large area around it that basically corresponded to the shape of the fungal patch, which is usually circular, and there would be a zone of inhibition around it where no bacteria would grow. And so he noticed that and realized the implications were that the fungal colony was making something that was inhibiting bacteria from growing. So key things: notice, realize the implications, and then he decided to sort of check it out in his spare time. That changed human health. That this was his discovery of this process has been probably the biggest benefit to human health in the past 200 years. And what he discovered as he kept working on this was that this fungus made a chemical that Fleming called penicillin. You would not say that Fleming developed penicillin. You should say that Fleming discovered penicillin, and then he went on to figure out how to purify it. And then after he figured out generally how to purify it, it could be chemically characterized and then made in laboratories and then sent to the world. So this was discovered in between the world wars. This is a cool picture. This is a fungal colony, which you can tell because it's fuzzy, and it's actually exuding or oozing out droplets of antimicrobial drugs on its own. We didn't make it do this. It does this on its own. So the very first antimicrobial drug that we knew about had existed for years. Because in the world of microbes, microbes make pretty numerous chemicals to fight other microbes. If a fungus can produce a chemical that prevents bacteria from growing around it, the fungus is going to have more access to nutrients and space and water and a more clear field in which to grow. Microbes fight each other with chemicals, and Fleming discovered that fact and then co-opted one of those chemicals to help us fight certain microbes as well. And he discovered this effect generally before World War II, and by World War II, penicillin was in mass production, and just that one antimicrobial drug saved so many lives of soldiers that were wounded on the battlefield. So in World War I, many people died, and many of them were from infections that they got after the original wound. So they rushed penicillin into production, made it available as soon as possible, and it saved many lives. And since that time, there have been development of many more antimicrobial drugs. And if you could magically develop the most perfect antimicrobial drug, it would have all of these qualities. The main one that's important is we want a drug to be toxic to microbes, but not us. There are a lot of ways to kill microbes. You can bleach them. You can kill them with alcohol. You can poison them with heavy metals. Those are not so good for killing them in our bodies. So we want them to be selectively toxic to microbes, but hopefully not harmful to us. We would like antimicrobial drugs to kill microbes and be microbicidal instead of microbistatic, which means just preventing from dividing or multiplying. So it would be better if the drug that we're using kills the microbes that are in us instead of just prevents them from dividing. Hopefully it will be soluble in body fluids and will be active in small amounts. These two are kind of the same to me. Soluble and active in body fluids. And hopefully active in, let's say, low concentration. If you've ever had to take some kind of antimicrobial drug pills that are huge, that one probably requires a higher amount to be active in the body. If you've ever taken a once-a-day tiny pill, that one remains active in the fluids and also is active in a smaller amount, probably. We hope it doesn't lead to resistance. I'll be talking about that next time. This one, if you make a drug and it's great and nobody can afford it, then what? That raises up a whole lot of ethical issues. You may have heard about that guy that bought a company that made some kind of anti-AIDS drug, and then he made the price high because he could. That's capitalism. Nobody cares if you do that with something else that's not a drug, but once it's a drug, then it's suddenly an ethical issue. That would be a good topic for economics. Why should he not? Why should he? Is that ethical? And hopefully the host will not be allergic to the drug, but are any of you allergic to penicillins? Poor you. There's a bunch of them. Highly useful, fairly cheap. Many people are allergic to them, which prevents them from being the perfect drug. There is no perfect drug that meets all of these characteristics. Some of them will have some of these characteristics, some of them will not. You may have noticed that most of the time I keep saying antimicrobial drug instead of the word antibiotic, because antibiotic scientifically has a very particular meaning, which is listed in this chart. If you as a science major or a science professional say something is an antibiotic, it is naturally produced by a microbe somewhere in the world. It has a naturally occurring source in nature. Antibiotic. So penicillin is an antibiotic because there are fungi out in nature that make it already. It is an antibiotic. When I am walking around in the world outside of Whitehall, I do say antibiotic because the general public, if I say antimicrobial drug, they're just going to be like, think that I'm snooty or something. But technically the term antibiotic means that there has to be, that item has to be found in nature somewhere, yes?
Are most the stuff we call antibiotics, like ampicillin or Antimicrobial, are those antibiotics? Or?
Some of them are, and some of them would be called semi-synthetic. Like I think ampicillin starts with penicillin as the base molecule, and then it's modified in the lab to be better in some way, and that would be a semi-synthetic. So these, the antibiotics, if they are in some ways boosted chemically in a lab, or they might be actually genetically engineered at some point in a lab to make some extra component that makes it a better drug, that would be a semi-synthetic. If a drug is completely synthetic, that would mean that it's made entirely in a lab. You weren't starting from any naturally derived products. You chemically synthesized that in a lab. And there are not that many totally synthetic drugs that we have that aren't based off of a naturally occurring antibiotic in the first place. One good thing about the term antimicrobials is that could cover all of it. Whether it's completely natural or it's semi-synthetic or synthetic, you could call all of them antimicrobials. These two terms, narrow spectrum and broad spectrum, these are referring to what range of microbes are affected by the drug. And we usually use these terms when we're talking about antibacterial drugs. Narrow spectrum would be maybe only a subset of gram-positives. Broad spectrum would be pretty good at killing gram-positives and gram-negatives, a wide range of microbes. And the other term I want to point out up here is prophylaxis. This is not a specific term just to microbiology or just to drugs, but prophylaxis means preventing. If you ever go on a mission trip and you take some kind of medicine before you go, that's not a vaccine, but you take some kind of medicines that are going to help prevent you from catching a disease, that's prophylactic chemotherapy. So some other terms that I want to sort of add to this, and they're pretty obvious what they mean, is antimicrobials covers it all. You're trying to kill a microbe, are you giving that chemical to a person? It's an antimicrobial. We can divide antimicrobials into antibacterials, and those are specifically targeting bacteria. Antifungals, antivirals, antihelminthics. These are your deworming medicines. Antihelminthics, like Ivermectin is an antihelminthic. And then there would also be a class of drugs for protozoans, but I don't think they're called antiprotozoans. And for some reason no one ever says that term, whatever it is. But These terms define what class of microbes is that drug targeted to. Out of this group, this set of drugs would be targeted toward eukaryotic machinery. As in, these class of drugs would be targeting either an organism that is already eukaryotic, like a fungus is a eukaryote, a worm, a helmet definitely is a eukaryote. Viruses that infect us are mainly using our machinery, and we are eukaryotes. So these are targeting eukaryotic machinery. These are usually targeting prokaryotic machinery. We are eukaryotes, not prokaryotes. And I'll come back to this topic in a minute, but this group of drugs tends to have greater side effects and are drugs that are highly not enjoyable. For example, the drugs that control foot fungus and toenail fungus, that's my least favorite commercial ever, where that little dermatophyte opens up the toenail and jumps out of it. You know the one? Those have to be taken for a long time, and if people have to take oral antifungals, it takes a long time to eradicate that stuff from under the toenail, and the side effects are, you will generally feel ill while you're taking them, because there are fewer targets that aren't shared when you're developing this class of drugs. Antivirals are also unpleasant to take. Anti-wormers are unpleasant to take. And we'll look at in a little while what are the specific targets and how similar are they to us. If you're not allergic to antibacterial drugs, they're usually pretty good, except if they interfere with your microbial flora. People getting diarrhea after taking antibacterial drugs, that's a flora effect, largely. And we'll talk about that after Thanksgiving as well. So, know the definitions and like how you would classify different drugs. So if I gave you a scenario about a drug that was developed in the lab against viral targets, you could say that it was an antiviral. It's synthetic. If it's only against a certain virus, it would be narrow spectrum. It would be an antimicrobial as well. So, thus far, antimicrobial drugs will have one of 5 general modes of action. And by modes of action, this is sort of saying, what do they attack? What is their mode of action? And each one of these, is this from you don't know yet. I don't remember which book this is from, but I like this picture because it's showing what are the classes, where are they acting, and what are some examples of drugs that use those modes of action. So this is a good study picture. A great thing to attack on microbes would be cell walls. And most of this list is attacking cell walls of bacteria. So cell walls of bacteria are made of peptidoglycan, which we do not have peptidoglycan. Attacking peptidoglycan would not have any direct effect on human metabolism. Fungal cell walls are made out largely of a polysaccharide called chitin. And I don't know if anyone has developed chitin inhibitors yet, but They should be working on it if they're not already. And notice how many drugs are in this list. There are a lot. Low side effects, specific to microbes, and these would tend to cause lysis. They would cause lysis of the organism by destroying their cell wall. The second category is attacking the cell membrane. The problem with this one is that we also have membranes, and the differences between, except for archaea, which can have some weird membranes, generally in life a membrane is a membrane and they kind of work the same way. Generally lipid bilayers, they're kind of similar throughout the world. So notice, this is not the only cell membrane attacker, but it's a generally shorter list. These would also disrupt human cell membranes. And this particular drug, polymyxin, you tend to find it in things that are applied externally. Like triple ointment, I think, has polymyxin in it, or neosporin. My family always just calls it neosporin, no matter if it's actually neosporin or not. So if you look on those little tubes of triple ointment, polymyxin is one of the things in it that attacks cell membranes. And there's probably a label on there saying don't put it, well, you don't put any of this stuff in your eye, but don't put it on mucous membranes because it would break those down. If you put it on the dead surface of your skin, that's not too bad. You don't have that many membranes sticking out there. So it's okay in that application. You wouldn't take it internally, unless you're trying to poison someone. Just say no to internal polymyxin. So cell membranes are not the best target. There are a few drugs that do target those. The next thing that antimicrobial drugs could target would be DNA and RNA synthesis, let's call it. DNA and RNA synthesis. And we have DNA and RNA, but fortunately, we do not have, that there are enzymes that relate to DNA and RNA that microbes have and that we do not. So, for example, there's one called gyrase. And gyrase inhibitors disrupt bacterial genome synthesis. If you remember that a lot of bacterial genomes are circular, when they copy their genome, they end up with two copies of genomes that are circles and they're together like that. Gyrase helps untangle them. We don't ever end up with tangled up circles of DNA, so we don't need gyrase. Gyrase disruptors would not affect us, so they're a good way to affect bacterial genome replication. And if you've ever been on ciprofloxacin, that is a broad spectrum antimicrobial because any bacterium that has circular genomes would be killed by it. When I went to Malaysia and got sick and they couldn't diagnose what I really had, I got ciprofloxacin and it was great. I got better. It's broad spectrum. There are some differences in microbial RNA polymerases that make them a little bit different from our polymerases that would provide a selective target. So rifampin, if you've heard of that drug, it inhibits RNA polymerases specifically and exploits a slight difference between ours and theirs, so to speak. You would think by now that we would have a drug against RNA-dependent RNA polymerases for flu. There are people working on them. They are not on the market at this point. But another excellent target, especially against bacteria, is to target bacterial ribosomes specifically. Bacterial ribosomes have the same general shape and the same large and small subunit structure, and also are made of small segments of ribosomal RNA, generally the same function, but there are enough differences in our ribosome shapes and their ribosome shapes that drugs can be specifically developed that inhibit bacterial ribosomes but not human. And there's a big list of these because they are specific and they are not toxic to us. They could either be acting on the large protein subunit, which is generally shown on the top of the ribosome. They could be acting on sort of the smaller segment, or sometimes the drugs are acting on the interface, the region in between the subunits, like they're touching them both, or they're in the cleft between the subunits. Big list, selectively toxic, often a good choice for bacterial infections. And then this fifth way, the fifth way is to block a specific biochemical pathway. To block a specific biochemical pathway that's only used by the microbe. In the laboratory, when we used the drug SXT, did we use SXT? We did in this class. There was one disc that we used on the Kirby-Bauer test that was SXT. That's a combination of two different drugs that block one biochemical pathway that is related to bacterial folate synthesis. Let me go and we used a P disc. Bacitracin you use for your unknowns. I used different ones in the two different labs this year, but some years we used tetracycline, gentamicin, erythromycin we did. We have an E disc. Included most of these classes. Oh yeah. And if you remember which ones were highly effective, ciprofloxacin was good against everything. And the others were kind of hit or miss which bacteria they were effective against. This is just showing specific places where specific drugs alter bacterial ribosomes. Whether it's small subunit or large, or in between the cleft. And this picture is showing one of those specific biosynthetic pathways. That was method 5. If you block this, that's method 5 of inhibiting specific bacteria. So remember the SXT drug, the S part is blocking this step, the T part is blocking this step. And so this would actually be a good biochem type question. You got pathway, block here, block there. The reason that those two are used in combination is that some bacteria have evolved resistance to one of them. If you include both, that's more effective to combat those bacteria. Great. Last slide for today. This chart is showing you the varieties of penicillin drugs. This is the one that was discovered by Fleming. It's called penicillin G. So the original. And it happens to be narrow spectrum. It's good against gram-positives only. Penicillin G is good against gram-positives only. And generally, as you're going down the list, you're looking at longer ago up till more recent. So down here are the more recent developments of these drugs, and penicillin G was the original. And then it tells you when would you use that, what are the disadvantages to it, and so on. So the penicillin class of drugs. And you can see that over time, there have been modifications to penicillins to make them more effective. Either they're absorbed better, so they cross through the intestine better, these two versions. Methicillin is more resistant to penicillinase enzymes that bacteria use to cut up these drugs. Ampicillin works also on gram-negatives. That was a big advance. Amoxicillin is the one that kids take for ear infections all the time. If you had a lot of ear infections and you took the pink syrup, it's amoxicillin. It's absorbed well and it works against gram-negatives and gram-positives, which makes it pretty good for ear infections, but also healthcare is moving away from giving kids early on lots of broad-spectrum antibiotics because of what it's doing to their gut flora. If you remember our Pseudomonas plates that were green and hardly anything worked against them, the newer varieties of penicillin drugs will work against them. These are too expensive to buy for our lab. The newer it is, the more expensive. The newer it is, the less likely you're able to get a generic. But if you saw me on Halloween and I was dressed up as MRSA, that stands for methicillin resistant Staph aureus. Where it says methicillin resistant, that indicates generally that it's resistant to everything above this line. Generally. Because these are still all penicillins. And methicillin resistant Staph aureus makes enzymes that will cut these up as a group. MRSA is worse than it sounds. It's not just resistant to methicillin, it's resistant to all of the penicillin group, basically. So that will be a good lead-in to after Thanksgiving when we talk about antimicrobial resistance.